everyone to join us here in the sanctuary. Our afternoon program is about to start. In 3 minutes, please come and join us. Our opening songs is Jesus is Coming Again. Number 213. Jesus is Coming Again. Come join us. Nori, come. Elsa, please. Marcia, you want to join us to sing the opening song?
Testing. Test. Checking, check.
Jesus is Coming Again. 213, first and last verses.
Trumpet and now let us sing. Jesus is coming again. Cheer up, ye pilgrims, be joyful and sing. Jesus is coming again. Coming again. Coming again. Jesus is coming. Last verse. Angry, by this we do know Jesus is coming again. Knowledge increases, and fro.
All right, testing. 1, 2, 3.
Jesus is coming again. Coming again. Please turn to next page. We have this hope. And we sing first verse only. We have this hope that bursts within our hearts. Hope in the coming of the Lord. We have this faith that Christ alone imparts. Faith in the promise of His word. We believe that time is near, and the nations shall awaken, shout and sing. Hallelujah, Christ is King. We have this hope that burns within our hearts. Oh, in the coming of the Lord.
Hello. Thank you. Last call, I would like to invite everyone from the fellowship hall to join us for this afternoon program. Let's have a prayer. Our most gracious, loving Heavenly Father, we thank you so much for your Sabbath day. We thank you for presiding over our divine service this morning. And now in the afternoon we have a help presentation. Please send thy Holy Spirit to be with our speaker, with the listener, so that the presenter will give us a good health message from you. And for the listener, we will learn something from the message today. In Jesus' name, amen. Good afternoon. It is my honor and privilege to introduce to you our speaker, Dr. Paul Kirshman. He is here today with his wife, Dr. Rosita. Dr. Paul received his BA degree from Cornell University in 1983, and his Doctor of Medicine in 1990 from the University of Montpellier, France. After graduation, he did a residency training in anatomical and clinical pathology at the Westchester County Medical Center, Walhalla, New York. From 1994 to 1997, he earned a fellowship in molecular pathology at the National Institute of Health, National Cancer Institute, under the direction of Dr. Sobel in Bethesda, Maryland. From 1997 to the present time, he worked for and works with a variety of specialized laboratories focusing on various diagnostic, prognostic, and therapeutic aspects of cancer and other pathological conditions. Paul, thank you for spending your time with us this afternoon to share your expert knowledge in certain aspects of medicine. Please, the time is yours.
Sure.
Thank you, Paul.
Thank you, everyone. Thank you for having me. Shabbat Shalom. I pray that what I'm trying to explain is easily understood and clear, and I hope Yahweh helps me in this endeavor. So today, I'm not going to make declarations or statements that everything I say is absolute, but rather present things in a way that make us ask the questions that have to be asked and to determine what the next steps should have been and must take place. And so today I'll be speaking about not probably what you're expecting given the headlines about DNA fragments of the plasmids used to develop the Pfizer-BioNTech vaccine against COVID. I'm going to talk about a problem that existed even at an earlier stage which wasn't addressed, and that's the fundamental problem of using messenger RNA as a vaccine. Without much ado, I don't have to remind you what these past 4 years were like. You know, we had the vaxxers, the warp speed, the big pharma industry hustling out new nanoparticles containing synthetic genetic sequences, and people wearing face masks and doing a whole bunch of religious rituals, which really didn't have much to do with science. And Ellen G. White, my understanding, because I read some of her writings a while ago, said not to put your faith and trust in politicians. And we need government for certain things, of course, and all, but we shouldn't trust in politicians, not any of them. And you have the old suspects here, and I was gung-ho for Trump at one point, but I hope he changes now and sees the light, but I don't trust any of them. So you had the vaxxers, you recognize at least two of the three people here. Fauci and Pope Francis worked very closely together. They had lots of meetings in Rome, calling in the World Economic Forum, the big pharma companies. I mean, they were like lovebirds. They were, talking to each other every week, And in any case, there were a lot of ugly slurs going around during this time. One of them, which sort of got me very angry, was the notion of anti-Semitism. Somehow, if you were opposed to the vaxxers, you were an anti-Semite. And so I kind of came up with my own theory about all this. If Vatican doctrine is correct, and if Pope Francis and the previous popes and the future popes are all the true Jesus Christ on earth, you know what that means? that means that Pope Francis is a Jew and a rabbi. We'll come back to this. So we have the rabbi in Rome who's very pro-vax. And I'm a Jew, and I've changed a lot, but I never thought he could be a Jew, but when I thought about it, who knows? So then you have the anti-vaxxers. Man, the neo-Nazis almost, the stormtroopers opposed to the vaxxers. And pay attention to the guys with the black hats in the background. Since, there were a lot of slurs and insulting going, especially one way. I want to show you these anti-vaxxers, the new anti-Semites, because they rejected the rabbi in Rome. Go figure, they were against it too, the ultra-Orthodox Jews, and, they couldn't call them anti-Semitic, but I guess if you go by Vatican doctrine, and with all the anti-Semitism supposedly amongst the anti-vaxxers, well, why not, And some more anti-vaxxers, they're wearing the star of rejecting the rabbi in Rome. But what people fear the most is being stigmatized because they refused the gene therapy. I don't think they're going to do something like make us wear yellow stars. I think they're going to try to be a little more creative. Maybe that for unvaccinated. I don't know if some of you know what that really is, but but who knows? It's a little more subtle. Or maybe we'll have to sew barcodes on. We'll have these QR codes on our suits or outer vestments so that they know who we are if we refuse mRNA. And you might laugh, but in Europe, most European countries, and in China and in Israel, people have to walk around showing their iPhones with the QR codes to go in and out of, restaurants, take the train, the bus, movie theaters, whatever. And so this is just giving you a little background, but I don't think you even really need it. But I just thought I'd address some of the ugly slurs going all around. And in any case, the main reason why I'm talking about this today is, going back, just a year or two ago, there were more and more headlines about the explosion of cancers amongst young people. And middle-aged people, and middle age is young today. More and more people were having cancers and more aggressive cancers than ever before. And it coincided within the year or two after the administration of the mRNA injections. And another headline, I'm sorry, I didn't read the other one. I don't know if you caught that, but the UK government data reveals young people are dying of cancer, explosive rates following COVID, years in vaccine push. And then Dr. McCulloch, a cardiologist, warns that getting jabbed multiple times with the mRNA COVID vaccines could promote turbo cancers. And I like that word because I don't want to divulge patient secrets, but I've had discussions with oncologists who've encountered turbo cancers related to certain therapeutic modes. Now, this was about 2 years ago. Something miraculous happened, a sentinel finding. A laboratory in Sweden, headed by Yang de Marinis, I believe I pronounced the name right, at Lund University in Sweden once again, they found that if they put a very low concentration of nanoparticles in tissue culture media in which a human liver cell carcinoma line was growing. These are human cells that proliferate on their own if you give them enough sugar and nutrients in the liquid on them. That after 6 hours they could find DNA copies of the BioNTech messenger RNA. And they didn't inject them with DNA, they injected them with mRNA, but they were finding copies of DNA in these cells only after 6 hours, multiple copies. And we'll talk about, everything I say will kind of overlap, with what I say now, but they also found elevated levels of what I'll explain later, a reverse transcriptase that's in humans called line 1, both in the nuclei of cells and in the cytoplasm of these cells. And that's how it was possible. And we'll get into more detail about that. Now, the United States, I just showed this picture of Neil Armstrong and his crew, the first astronauts on the moon, just to remind us that our country used to take the lead in things. We were almost always the first to do different things. We had Lindbergh be the first transatlantic flight. We had the first astronauts on the moon. We used to take the lead. But Yainda de Marinis at Lund University, it's not the most famous place in the world, with her fellow researchers in Sweden, did something equal to landing on the moon. They demonstrated that the messenger RNA, the synthetic messenger RNA of BioNTech, can be copied into DNA and put into our cells' nuclei, something that we were never warned about. And the manufacturers, I can't believe they didn't know the possibility of that happening. And I show this picture too because I might not have the same emotional attachment I do to the subject, but if it were up to me, Yainda Marinis in Sweden and her members should have a photo like that with their heads on the equivalent of astronaut, in astronaut costumes, because to me and to the rest of the world, they've done something equal to landing on the moon for the first time. And we'll get into that a little more. Now, when I was looking at the sequences of BioNTech and Moderna, of course, the findings was with BioNTech made by Pfizer, but Moderna is supposed to be the copy, almost the same thing, and there are nuances and differences. But when I was trying to research for myself, get information about the sequences for these and compare them to the virus, I came across this abbreviation ORF. And if anyone's from the 70s here, they may have watched ALF. ALF, that kind of like dog-like alien who lived with a family. It was a comedy, and he used to love to chase and eat cats. But I guess I'm showing my age. Maybe no one else has heard of ALF. But that's what I thought of when I saw this abbreviation ORF. And what does ORF stand for? It stands for Open Reading Frames. So I apologize that this writing is really tiny, but this is a lineup, same size and everything, of the wild type, that is the messenger RNA in the COVID virus, the BioNTech BNT162, that's the Pfizer mRNA, and this is the Moderna mRNA 1273, and they're all the same size. And we're going to come back to why I'm talking about this, and it'll hopefully click in your brains a little later. But you see all these ORFs, and I apologize, it's really tiny, all these different proteins. Now, in viral messenger RNA, unlike our human RNA and mammalian mRNA, and even the mRNA in fish and other more advanced species, there is this ability for the same strand of mRNA to code for different proteins. One mRNA can code for lots of things, the outside of the protective code of the virus, inside enzymes necessary for division, for rather copying the mRNA, for transcription of the mRNA in the case of an mRNA virus. I apologize that you can't see it from where you're sitting, even if you're up front, because the print is really tiny. But we're going to come back to this. One of the things I noticed in the wild type, these are all the same size, all to the same scale. There were several ORF1 sequences all along the length of the viral mRNA, the BioNTech mRNA. These are all ORF1s, all these intermittent stretches. This means that in addition to the S protein, to the protective capsule protein, to the various enzymes, they have this open reading frame, and you're going to see why it's weird. And this is why I think I hope she'll never yell at me or say I'm wrong, but I believe that this is what sparked her thought, that this is ORF1 too. The whole length, you have these ORF1s, the full length of each one of these, the wild type, BioNTech, and Moderna. And we'll come back to why that's important, but this is why I think she did her research in part. This is, are there hidden genes in DNA, mRNA vaccines? And this is something I basically cut and pasted and changed a little out of an article. The presence of putative open reading frames, ORF, on DNA mRNA vaccine candidates implies that overlapping genes may contribute to the translation of smaller peptides, building blocks of proteins, that is, potentially leading to unintended clinical outcomes, and that the protein coding potential of DNA mRNA vaccines should be rigorously examined prior to administration. So this is out of a publication in 2022. It's 2024 now. This is what the Lund group figure it out. They figured, they had this suspicion, and the reason why they have this suspicion, and I believe I show it in another Kodachrome later on, there was this article that scared the hell out of everyone in China. And they were saying how the viral mRNA of the virus, the COVID virus, was being copied into DNA and inserted all over the place into the genes of humans. And that was supposed to scare everyone, run out and get the vaccines because you don't want to have COVID. And I think I'll end up repeating myself again, but she was saying, what if the vaccine mRNA behaved the same way as the viral mRNA? And so what they did in this experiment, and by the way, just details on the side, this is the construct of a messenger RNA in general. You have the untranslated regions, that means they're not translated into protein. You have a signal area which tells the ribosomes where to settle down and to start reading the mRNA to make proteins. Then you have an untranslated region on that end, UTR, and then along what's called a polyadenosine tail. The purposes of the stuff at either end is to stabilize the mRNA so that it doesn't degrade too fast. And the transcriptions takes place along the coding sequences. And the BioNTech mRNA, just like the Moderna mRNA, are synthetic. They're made in a lab, and they do this for a number of reasons. But the team in Lund, Sweden, realized that there was something very proprietary, very uniquely present in the BioNTech mRNA that constituted what this untranslated region actually corresponded to a human mitochondrial DNA, then the 3UR there in green, and the artificial spike protein, and it's artificial, and I can hopefully say why a little later and we'll remember, this was representative of the spike protein in the virus, but mutated. It's not the same thing. But there is no reason why the spike protein and human mitochondrial DNA should be in the same stretch of a messenger RNA. If you don't understand the concepts yet, I'll give you some basics shortly. But this was something uniquely proprietary, if you'd like, to the BioNTech sequence. And what they did, and I'll explain it a little later, what it means exactly, they designed primers to flank this area and to amplify, this unique stretch, and we'll get into what bases mean in a moment, but this very unique proprietary stretch to identify DNA in the cells after the incubation with the BioNTech nanoparticles containing this mRNA. And what they did is they put in an enzyme that, after 6 hours of incubation, and they did different concentrations and different time frames, but 6 hours is pretty short. They broke down all the messenger RNA so it couldn't possibly interfere with the PCR, and they amplified lots of copies of this unique sequence, proving that it can only have been for the messenger RNA. The only way you can have DNA in a cell that has this S protein, the altered, the proprietary S protein with the human mitochondrial DNA attached to each other in a sequence, which they did, could have only been from the BioNTech messenger RNA. And if you don't follow me yet, I hope it'll be a little clearer later on. So I'll give you a little background. This line 1, it's to help you understand the significance of line 1, and I'll tell you what it stands for in a moment. Only 1% of our DNA is recognizable as protein coding genes, and that's they're in the sequences called exons. But the thing, the take-home message is only about 1%, a little more, some say 2% now, of our genetic code is understood. The rest is a whole mystery and speculation and whatnot. So we're only scratching the surface of our understanding of our own genes. And much of the intervening sequences are intronic. They don't know what it does. In other words, intergenic between genes. And also little remains of mobile DNAs that get duplicated and inserted sort of randomly. And they're trying to understand why that happens. And transposable elements, which we'll come back to. Line 1 is a transposable element. And transposable elements, there's some that copy DNA of a cell and put it back in elsewhere, and then there are retrotransposons that copy messenger RNA of cells and put them back in somewhere in our genes. And in any case, we carry hundreds of thousands of copies of these sequences scattered as interspersed repeats, and collectively they make up half of our DNA. So, long interspersed element 1, line 1, that's what line 1 stands for, or L1. They are basically, try to simplify these sentences here, they basically take RNA in the cytoplasm, bring them into the nucleus of a cell, copy the RNA into DNA, and insert them, pretty much haphazardly. In this transposable element line 1, we only have one kind, line 1, but other species have several versions of them. And we have trace amounts of the other, but they clinically don't matter. And plants, they're really heavily loaded up with lots of linear interspersed elements, and we'll get how this was all discovered in a moment. But plants do a lot of copying of their messenger RNA and stick it back into their genes, much more so than animals. And simpler animals like mice do it a lot more than humans. And we're only beginning to understand, the full reasoning for that. But this is stuff that was known for a very long time, going back to before the 80s. And once again, only 1%, some will say 2%, of our DNA has a known function. And 17% of our known genes is a linear intertransposed linear interspersed linear interspersed elements, line 1. 17%, of which about 5 to 6% are also line 2 and line 3. That's much more prevalent. You have all 3 prevalent in all species, but line 2 and line 3 don't play a role in our development. And so, yeah, all retrotransposition is driven by line 1. And intact line 1 sequences measure about 6 that's a long stretch of gene, a little like the vaccine you saw there, the vaccine mRNA, but longer and in DNA. Line 1 is composed of open reading frame 1 protein and open reading frame 2 protein. And they also have some additional proteins that ensure that the copying of the mRNA is accurate when it gets copied into a DNA. But basically, and we'll see diagrams to help you, but open reading frame 1 protein basically is the transporter of the messenger RNA. It binds, sometimes a few of these ORF1 proteins bind to the RNA, brings it into the nucleus, then the ORF2, once it's in the nucleus, takes care of copying the mRNA and splicing it into our genes. And so, there are approximately, this is the article it came from, but there are approximately 5,000 line 1 copies in our human genome, once again composing 70% of our human DNA. That's a lot. And however, the majority of line 1 retrotransposition competency is well, I won't get into that, but basically it just explains how it works. And I put in red the much more important part of it. It's estimated that the average genome contains 50 to 120 active line 1 genes, and a highly active subset, which is about 5 to 10% of the active elements, and they're called hot line 1s. So this is what we have in our cells. They knew our cells could take mRNA into the nucleus, copy, and splice it in. This has been known for some time, and yet they have these sequences, BioNTech, Moderna, and the virus itself, that have all these open reading frame 1s, and I'll get even more into why that's important. And it's believed that the line 1s have a role in reshaping our genome, sometimes for survival reasons. And that's why plants have so much more activity. Plants are so much more complicated than we are in many respects, and they shuffle around their genes and copy their genes and insert them all over the place. And that's why you have the mustard seed, for example. There's so many variations of mustard, and I think broccoli is even one, and they look so different, and they even have different number of chromosomes, which we'll get to in a moment. But these line 1s also can participate in the pathogenesis, that is the evolution, the creation of many genetic diseases, including cancer, because sometimes the copies of the mRNA can be inserted in the wrong place and promote a cancer or suppress a tumor suppressor. They can screw up metabolism. They can screw up immune cells strengths by weakening them through various mechanisms. So depending on where copies of DNA are inserted, it's sort of like throwing a monkey wrench into an engine. Or it could be beneficial too. I mean, it's not always bad. And so here's some background stuff. There's no quiz and nobody has to memorize any of that. But I thought it was important I give you just a brief idea of how our genes are made up. And basically, they're made up of pairings of purines and pyrimidines. Those are nucleic acids. Our DNA is called deoxyribonucleic acid because they're amides which have sugar and phosphate groups attached to them. And sorry about that. And, these are they're basically the building blocks are adenine, thymine, cytosine, guanine in DNA. And you don't have to memorize any of this. It's just so I say it. And then in mRNA, which I'll get to in a moment, the thymine is replaced by uracil. And DNA basically has our code in interspersed non-coding areas that form double helixes, and RNA is single-stranded. DNA has the code for the RNA, and RNA is what's read by what's called ribosomes to generate all the proteins we need for a myriad of enzymes and cell division regulators, enzymes that are metabolic and enzymes that are involved in copying DNA, enzymes involved in the translation of RNA into proteins and organelles that are responsible for making our proteins and all. But this is just to show you don't have to memorize any of this, but these are the components of our nucleic acid sequences, our code. And our codes, I don't want to spend too much time on this, but I put this in here because there's a difference between naturally occurring mRNA in our bodies and artificially created mRNAs in the mRNA vaccines, and that's because the manufacturers of the vaccine mRNAs want them to last a very long time. And their way of doing that is treating one of the nucleotides, uridine, in messenger RNA in a way that sort of twists around one of the amide rings into not quite a mirror image, but into something that doesn't break down as fast and that resists degradation by our enzymes that break down RNA. RNA normally only lasts about 2 to 4 hours on average for most proteins, and it's quickly broken down. So the manufacturers wanted to make something that'll last a long time, and now we're hearing that it's lasting 2-3 months. I mean, they're just lasting forever, and they're not being gotten rid of the way normal RNA is gotten rid of, which poses risks, both in terms of they're constantly making proteins that they should have stopped making, and also making them much more susceptible to being copied by line 1, which takes messenger RNA and puts it back into the DNA as copies of the mRNA into DNA. And well, I'll repeat this again earlier. This is just to give you a background. You remember I was talking about all those open reading frame ones? Well, line 1 prefers copying itself, and so all those open reading frame ones needs to be explained, because to me it seems like an open runway for the line 1 retrotransponson enzymes to attack and move along and copy, because they prefer capturing themselves and putting themselves back in the nuclei. So why all those open reading frame ones on Moderna and BioNTech remain to be explained. But getting back to stabilizing the RNA so it lasts a really long time, this just shows you in this picture, I'm not going to bore you with all the various ways that RNA is broken down, but normal RNA generally doesn't last more than 2 to 4 hours. BioNTech, and Moderna, they have the pseudouridine, and we don't even know how long they're lasting, but they seem to be lasting a long time and showing up all over the place, including in ovaries, testes, brain, vessel walls. Somehow that RNA kind of likes to stick around after they modify the uridines into pseudouridine. And let's see. Oh, sorry, I skipped something. These are the two people who got the Nobel Prize in Sweden in 2023, pretty recently. But this is a picture when they got a prize a year earlier in Japan. And by the way, it really broke my heart when I learned that these two people got the prize for what they did, considering all the problems that we're having and all the uncertainty and all, because I think it should have gone to Yang de Damaris, who really, proved something we haven't addressed yet in terms of the safety of these messenger RNAs. But these two people got the Nobel Prize for stabilizing the RNA of BioNTech and Moderna. And I had a Walter Weiss moment when I came across these pictures, which is why I put it here, because you may note this and this. This is Japan a year earlier. And of course they're wearing masks, even though they've been working together for, like a decade or more than that, I think. So obviously, they're probably not married, or they're hypochondriac lovers. But this little thing here bothered me a lot, And maybe I watched too much Walter Weiss, but then I started thinking, where else have I seen these symbols? Here, there, I don't know if you can see it. I don't know what all this means. In any case, normal DNA has, don't strain your brain trying to remember anything here, but what I was trying to illustrate is that for each amino acid, which is the components of our proteins that we need, there's a redundancy for, I think, almost all of them, an alternate triple base codes. Everything in our genomic code is 3 nucleotides at a time, all strung in consecutive order. And it's, generally speaking, well regulated in our cells, but sometimes things go wrong. But there is this redundancy of the code in the DNA, which reflects itself as a redundancy in the complementary bases of the mRNA, because mRNA has to be made off a DNA template. And this is just to show you that these triple nucleotide codes, there's a redundancy for I think all of the amino acids. So a different triple code, could code for the same thing depending on the RNA, its length, a whole bunch of other properties, which also opens the opportunity to changing the triple codes for proprietary reasons, because you cannot get a copyright on wild type anything that occurs in nature. That's what wild type means. You can't get a copyright or patent on. But if you start changing around the triple codes to different triple codes that are not the same sequences in the wild type, the one that occurs in nature, you can get patents. So this is what Moderna and Pfizer were doing. They were putting pseudo-uridines that slow down the aging, and that's part of a patent. And they were putting alternate triple codes when they made the messenger RNA, so that they can say it's not wild type. We created it in a lab, therefore we have a right to a patent, because otherwise you couldn't make money on it, because you can't copyright or patent any natural occurring sequences since I think it was 2013. And they tried even before that, because they had the sequences already more than 10 years ago. So this is just to give you an idea and now this is like a cartoon version of what our DNA looks like. Just so you have a general background. And what you have is these complementary base pairs. This is like opening it up so you can see it better. All binding A to T, C to G, A to T, G to C, C to G, C to G, etc. And our DNA strands are very, very long. And go on, from our perspective, what we would feel like is an infinite length. So I won't dive too much into this, but, I showed the cartoon images of some of these. But nucleotides are formed from ribose, nucleobase, and organic phosphate, and all that's, how are nucleotides first formed? Oh, this is why I put this frame here. I know I'm not doing this justice, but for all these people who think that life occurred spontaneously, these sequences, and you'll see again in later projections, there's so much that has to go right for life to take place. Codes, there's so many proteins and there are transcription factors, enzymes, so much involved in replication of DNA, and you'd ask yourself, where do nucleotides come from? Where do amino acids come from? Which came first? You can't have the amino acids. You can't have proteins, in any case, without RNA, and you can't have RNA without. Well, you could have RNA without the DNA, but to get a code, How did that happen when you need enzymes to make RNA, you need enzymes to make DNA, and enzymes are super duper complex proteins that depend on nucleotide sequences, and then there are transcription factors. There are a myriad of things going on, and maybe I'm not explaining it well, but this is a highly complex system. So when people think that lightning struck a pond of salty water with some ammonia and oxygen and CO2 in it or something, and this just happened spontaneously, is really a stretch. In any case, this is just some background information, which I thought was fun. We have 23 chromosome pairs, total of 46 in a normal person, and we have 20 to 40,000 functional genes. We have about 3 billion base pairs. It's what I show you, the scale and the ladder, the steps on the ladder. The DNA in each chromosome, I'm just reminding you, is double-stranded, and each gene is composed of one long, super long stretch of DNA, knowing that the genes constitute a bit more than 1%. Our genes, our known genes, constitute 1% of the total billions of base pairs we have. Is still kind of like saying, it's that we only know very little so far about our genome. There's still so much more to be discovered. And this is just some conceptual stuff I threw in there. But many of you may have seen, and maybe in school or in college or something, when you had to take biology or high school, you may have heard of karyotypes. And this is, we have a copy of each chromosome from our parents. One from the mom, one from the dad. You can't tell by looking at them, but we have 2 chromosomes, one from each of our parents, for the 22 autosomes, and an X chromosome and a Y chromosome from, our mom or dad. And that's what makes us a man or a woman. If we have the Y, we're, Y and the X, we're a man. If we're a woman, we have XX, and if sometimes you have extra copies, but those are rare, but it does happen, and those are pathologic states. And this is how our chromosomes would look. This is a fake picture of the karyotype, because they're not condensed like this unless the cell's in division. And I just thought I just threw it in there. But this is, when you see all these X's, X's, X, 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 that's like the picture I just showed you. But you can only see them condensed like that when the cell's about to divide. So you actually see 2 copies of every pair of chromosomes. A pair of dividing one chromosomes from the father or the mother, you can't tell in the microscope, and one from the mother or the father dividing here. So you usually see double copies of each chromosome when you look at a classic photo of a karyotype. I just thought I threw that in there. But here's the cool stuff. This is like a cartoon representation of double-stranded DNA in a chromosome about to divide, but each half is equal, like a mirror image of the other, essentially. And this is, it doesn't look like that. It's much more condensed and dense, like in those photos. But this is how our genes look when they're in our nuclei in a resting cell, making protein and at rest, a cell that's not going to divide. The single strands spread out like the ramen soup noodles or something. They're just all over the place, spread out. They're not thick like that, but I'm just saying that they're spread out like a soup in the nucleus. They're not condensed like in those pretty pictures, because they're needed for all kinds of reasons, because all kinds of copying into messenger RNA is taking place all along them for all the different things a cell needs to be alive. So this is, when you look through a microscope, and you look at a cell nucleus, you don't see spaghetti. It just kind of looks like a blue, like a haze or something that's darker than the cytoplasm that is the outside of the nucleus. And it's just and once again, this is to remind you what a single strand looks like if you kind of stretched it out. And what I found fascinating, I was looking up some statistics. If you would take all the, we'll get to this in a moment, if you take all the DNA in one single nucleus of a cell, which is really tiny, and you need a microscope to see a cell. You're not going to see it in the air or on a table, or, you need a microscope to see one single cell. And the nucleus is often smaller than the rest of the cell around it. But if you take one single human cell, and you would stretch out if all the DNA could be, one continuous chain, like one chromosome's DNA linking to another, to another, to another, it would stretch out about 2 meters, or 2 yards to 2 meters. And you say, well, 2 yards, whatever, 2 meters, that's no big deal. But if each one of these base pairs here, if these if this GC was 1 millimeter, let's see each of these little rings in the ladder were 1 millimeter apart. This is what's really cool. If you could make each one of our strands of DNA from each chromosome, our 46 chromosomes spread out as a long string, it would be, just think of this as being 1 millimeter, it would go 7,000 times the height of the Empire State Building. So just to give you an idea how complicated we are, this is a miracle that we have all this code and non-code and stuff we don't fully understand yet compacted into every single one of our cells. And it's almost like every single one of our cells is a highly complicated biological computer. There's a lot going on a scale that boggles the mind, just like the ends of the universe boggle our minds if you think about it. And so this is just a little reminder, just so you know. You have your DNA. RNA is copied off of the DNA. That's called transcription. So the code is in the DNA. RNA reflects that code to a T with the substitution of U for thymidine. You don't have to memorize that or anything. mRNA goes out in the cytoplasm where the proteins are made. You have ribosomes reading the mRNA. Ribosomes have to read the BioNTech mRNA. Ribosomes have to read the wild type, the viral RNA. Ribosomes have to read the Moderna RNA. So basically, ribosomes are what adds what they call, what they call transport RNA, tRNA, which has respective amino acids, based on the codes matching the codes, those triple codons along the lengths of the RNA, and basically they string amino acids into what they call peptide chains, and then proteins are made up of multiple peptide chains, and where they get put together by other enzymes into complex proteins with various functions. Just to give you a little background, and maybe I didn't make it perfectly clear, and this is just to remind myself to tell you that a lot of this, most of what they call translation, that is the making of the proteins, takes place on the surface of what's called the endoplasmic reticulum, and a lot of the proteins we make get sort of spit through little pores in this endoplasmic reticulum into this empty space where they get processed further, and some of them get transferred to something called the Golgi apparatus, which looks like that, but puts it out into the cytoplasm so that hormones, stuff like that one cell makes can make it to another cell. Some of the proteins stay within the cell and don't go out of the cell, and those tend to be in the cytoplasm without the assistance of that. And some go back into the nucleus, proteins that are needed for DNA replication, for repairing DNA, for RNA transcription, and then the transcription factors and all kinds of things that go back and forth from the outside of the nucleus into the nucleus and to the outside circulatory system. I'm just throwing that in there. Now, I told you earlier, and let me just see. Okay. I told you about that 444 base pair proprietary sequence belonging to BioNTech. And I thought that coming back to that, now that you had a little exposure to DNA and RNA and what sequences are and all, I just wanted to briefly explain what they call PCR, and I'll try to make it easy. But basically, you target a sequence that you want to amplify with complementary, what they call primers that hybridize, and I think it's even, that hybridize, you don't even have to strain your brain too much on this, but that hybridize to the sequence of interest. And it's actually too small. That's the problem. I apologize. But you have primers that bind to one end of the sequence in one direction, because don't forget they're double strands. Primers that go in the other direction. And basically what happens is they start copying each other, and it gets to the point where the primers, and this doesn't show, I'm not doing a good job, but in the interest of time, you end up generating very surgically the same length you're targeting because of the possibilities of the primers annealing. It's the smallest that grow fastest. And you start making the specific target in much greater amounts, which I think is better shown here. But, you duplicate in the first round of duplication, you double, and you double what you doubled, and double what you doubled, and you get exponential amounts of the targeted DNA. And I apologize for maybe not making that clear, but let's just say after, this is 1, 2, 3, I think 4, 5, after 5, 1, 2, 3, 4. After 4 cycles, you get one 173,741,824,000. Oh, I'm sorry, after 30 cycles. This is usually a PCR reaction is 30 cycles. I apologize. After 30 cycles, which they didn't show here, but they're more, you end up taking a target, a small target, and amplifying it 100 million times. And so if you were to look for that 444 base pair, odds are you won't miss it even if there's a very low copy number. And what you do is you run a gel, and in this case it's 500 base pairs, it's just an example. You see where that's the ladder, you see where your product falls. This is from something completely unrelated, but you run a gel, that is, you put the DNA and label it with a fluorescent dye and run it from a negative to a positive charge, and it separates based on size, and the smallest will go the furthest, and the heaviest will move the least, and you put a ladder, a scale with known sizes, and you look to see if you have your product. And basically, this is just to give you an idea. You can fish stuff out of something very complicated, a tiny sequence out of a whole vast range of sequences, but this is just a chromosome map. I couldn't find a good one from a human. I think it's a different species, but it's just to let you know that geneticists work with these maps, interrelating what we do know, that 1% to 2%, and how they interact with each other, and all these different circumferential colors represent different characteristics of the various genes, their location and whatnot. So this looks really complicated. But if you were to put it on the scale of what we know and we don't know, what we know would probably fit in something about the size of that little yellow box. And so the rest is still the unknown. And we call it junk DNA until we find out it isn't junk DNA, and every day they're discovering that more and more of what we don't know is not necessarily junk. And getting back to the jumping genes in line 1, actually back in the 30s and 40s, this scientist McClintock was looking at, she was asking herself why Indian corn grows different than the stuff we're used to eating, and why you have kernels of different colors. And she started hypothesizing that whereas just sort of translating it to humans, whereas we both come from our mother and father, and we both we get half our chromosomes from our mom and half from our dad, and our cells are kind of equal to each other with respect to the number of chromosomes we have. As they differentiate, things get turned on and off in cells, and extra copies of some genes are made by retrotransposons and also by copying DNA in the nucleus and inserting it, so that the cells that are like our colon cells, our skin cells, our renal cells, our kidney cells, they're not all equal. They have different copy numbers of genes. And she was speculating back then that there must be something turning on and off these genes, so why some corn kernels are pigmented, others are not. And that's when this concept of the jumping gene hypothesis came about, and when the concept of transposons and retrotransposons came about. And the same thing with flowers. And there were studies in Japan, and there are about 6 different genes, and they're basically processed sort of in a row that determine these the colors of these plants. And these are whatever morning glory flowers, and I think the ones in the Southwest are speckled and spotted. Now, all that has to do with jumping genes, retrotransposons, and transponsons. So this stuff is known and real, and people have understood that both our genes in our cells, as they differentiate, can be copied in the nucleus itself and inserted in other places to turn on and off genes, to have extra copies. And the same with the RNA that gets out of the nucleus can be brought back into the nucleus and copied into DNA and inserted. And this contributes to different behavior of different cell types in our body. And so I come back to this just to repeat myself, and also hopefully give you a better understanding. We have active or hot line 1 copies in locations that are genes that vary from person to person. We're all different in where our active hotspot line 1s are in any given cell, and from individual to individual. There are many factors governing the activity of line 1, and it's a vast topic. And I won't get into methylation and all, it's a big topic, but methylation is a way of turning off and on genes as well, and the removal of methyl groups that usually activates them. So once again, you remember we had a Dr. Schaffenberg talk about the importance of exercise. This was like 2-3 months ago. Well, I just threw this in there because line 1 is, as we age, and the less activity we have, line 1, open reading frame 1 messenger RNA is higher in older humans, but if an older human starts exercising, it goes down. What it all means, I have to admit, I didn't quite fully understand it, but it seems to go in line with Dr. Schaffner's talk about how exercise, regular activity, and even someone overweight who's aging, that person will probably live more than someone who's normal weight or skinny is sedentary, especially as they get older. I'm showing this just to show you how back people were aware of line 1. It's changing topics slightly and getting back to what I thought was most important. But look at these dates. You don't have to read the whole text here. 1984, 1986, 1981, 2000. People knew about line 1, and they knew what it did. It takes RNA, copies it into DNA, and inserts it into our genes. And yet, this is something, and maybe I should have shown it sooner, but I remember I mentioned to you that there was a group in China that published a study showing how the COVID mRNA is copied and inserted into our genes. Terrifying. And they went so far as to make a map of the, kinds of, or rather a pie chart of where they found them. And so they were doing a study like Yang de Damaris in Sweden, but they were studying COVID, which was okay. They weren't shut down the way she was. But they showed that the chimera, these are where there's chimera means hybrid between the human sequence and the COVID sequence, where they were inserted and they don't belong, giving chimeras. And they were showing that these are just 2 different sequencing methods. That's when you determine the sequences, which I'll talk about briefly in a moment. When you sequence them, it tells you what the sequences and the codes I was telling you, telling you about the stretched out codes. It reads it base by base and spits out what the sequences are. Well, going by the sequences, they could tell what was upstream and downstream from where the COVID mRNA was inserted. And some of them were inserted in exons. This is just 2 different methods, and methods could change a little, skew results one way or the other, but the importance of the findings are in these pie charts. About 23 to 28% were found in coding sequences that were necessary for the proteins. It didn't go into the consequences, but they were mutating mRNA. They were mutating sequences that would lead to mutated RNA and the downstream consequences of that. Some of them were in intergenic spaces, 23 to 28%, meaning they don't know what it is. And then introns are these sequences, which I didn't show you, but between exons, there are introns which get spliced out when you get the final mRNA. I just try to make it really simple. But they were proving that COVID mRNA gets inserted into our genome, which is why Yang de Damaris and her team said, let's just see if the BioNTech, they picked that one, I hope they work on Moderna one day, but it isn't likely. Let's just see if you could say the same thing about the mRNA vaccines as you can say about the COVID virus mRNA. And they were in the steps of, they took the first step and demonstrated it was there, but nobody has followed through mapping out where it is by sequencing, which has to be done. Instead of people saying that she only did it in vitro and saying that she didn't, do it on more samples, she was shut down and she can't do it. But they let the guys in China prove all that the mRNA got inserted all over the place, basically, from the COVID virus itself. And this is just a map of, and the reason why I'm showing this is that as a pathologist, I have my suspicions where the synthetic mRNA strands, the nanoparticle mRNAs or the mRNA vaccine mRNAs can be inserted. And there are tissues in our body, the cancers in the tissues in our body that have high line 1 activity, which means that if there's a lot of line 1, it could pull in other RNA and throw it into the genome of the cancer cells nuclei, our cancer cells genes. And breast has a super high Liver is lower. Colon's, about halfway on the scale of having line 1 activity. Ovarian has a lot of this retrotransponsin activity, line 1. Lung about halfway. Esophageal cancer has more than some of the others. Pancreatic has very high line 1 activity, and prostate cancer is a little lower in line 1 activity. And I hope I'm making myself comprehensible, but I'm sorry. Someone wanted to say something? Oh, but in any case, I believe that the cancers, if you have a cancer in you, and let's say it's in its early stages, but it has a propensity to have high line 1 activity, it would be more likely to uptake BioNTech or Moderna mRNA and stick it into the genes, which could possibly accelerate the cancers. And if you had a cancer that might, this is something you don't hear much, but we all have, especially in our colons, but we have sometimes micro cancers that we just shed out, a little polyp or something with a little precancerous or a little micro-invasion cancer, and we basically excrete it. And in other parts of our bodies, we have moles and stuff that become melanoma, but then the inflammation makes it disappear. And then, of course, we have, people who have very early cancer, and we don't know when it's really early sometimes. And my big concern is, you've heard about turbo cancer and all. Nobody is looking for the possibility that these RNAs are being copied and spliced into the genes of the cancers we have, possibly making them accelerate. I say possibly, although I have my suspicions. Nobody has ever studied the oncogenesis, the ability of these things to cause or not cause cancer, nor are they screening for the proprietary sequences that could only come from these nanoparticle, these mRNA vaccines. There's just complete shutdown, and they mock the people who are questioning this, although I found a few excellent articles, contemporary articles, explaining the risks of this, and nobody wants to pursue this because nobody's going to give grants for this. It'll cost a lot. Not as much as you'd think, though. There are ways of doing this very economically, but they should be searching for the possibility of these RNAs being incorporated into our DNA. They already are saying the contaminating plasmid DNA. Plasmids are like circular DNAs in which they insert DNA sequences to make the RNA artificially. Well, apparently they didn't clean the plasmids grow in bacteria. They're a form of circular viral RNA. They didn't get rid of that DNA, and now they're saying that DNA is being spliced into our genes. And I wasn't, before that ever came about, I was saying to myself, why aren't they proving that the mRNA itself is not being copied into DNA and inserted into our genes, possibly causing cancer, possibly causing lots of other problems, metabolic, inflammatory, so what did I put this here for? Just a take-home message, ORF1, the protein, is a chaperone protein. ORF2 is what copies and splices the RNA into DNA and inserts to our sequence. These open reading frames, as I say, tend to favor themselves. They like copying their own RNA, which brings me back to the ORF1 stretch of sequences. Why are there so many ORF1 sequences in the BioNTech and in the Moderna RNA? I don't know, but they have a tendency to prefer to copy themselves, and that seems like an invitation to the line 1 to say, hey, come here and copy me, put me in your DNA. And I've spoken to geneticists. Nobody can explain to me why there are all those open reading frame 1s on BioNTech and Moderna, and of course it's in the wild type. So that's, this is just I may be being redundant, but I just wanted to remind you of the function. So, you have line 1 messenger RNA. It gives you the open reading frame, which is one part of the product, and open reading frame 2. They go out and bind to another RNA, usually itself, but it could be another RNA, bring it back into the nucleus. This is the chaperone protein. That's the reverse transcriptase and splicing protein, and it inserts it in a pretty cool way, which I won't get into, but it finds sequences that would like to take in the poly A tail because, but not necessarily bind to all of it, but even some of it to give it like an anchor. It splices open our DNA wherever it sees a few T's in a row and inserts in the DNA that it copied from the RNA. And this is old stuff. They knew this stuff 20 years ago. And so my thoughts, and this was about a year ago, was one of the first steps a serious lab that wants to get to the bottom of things would do is duplicate the research in Lund, Sweden, and look at in cancers, especially, like you remember that the pie charts I showed you. In cancers known to have high line 1 activity, why not take actual human samples? And you could do this on old tissue, paraffin-embedded blocks, which is like the standard practice in pathology. You can extract the DNA and do a simple PCR test looking for the BioNTech proprietary patented sequence, and if it were there, the next thing you could do is submit it for what they love to call, it's very fancy sounding, next generation sequencing. And by sequencing the genes of a patient who has positive PCR test, you can tell wherever it's been spliced, if it's in an area that might cause some metabolic problem, in an area that might activate an oncogene or shut off a tumor suppressor gene, if it's inserted into nowhere and has no consequences, in which case it means it's basically tattooing your DNA. I believe that we have to start looking for this in all these so-called turbo cancers where patients had explosive cancer right after getting either the BioNTech or Moderna. Now, the PCR, the one in the article is very specific, and in time you could find other specific targets. You can't expect just one university to do everything, especially when there was so much pressure for them to shut down this research, and they've stopped. And I can't find any follow-up. And a lot of people were saying, oh, she only did it on human cell lines. They had no problem with the Chinese research proving COVID was copied into DNA and inserted all over the place, but they won't find out if the BioNTech and Moderna mRNAs are doing the exact same thing, because there are a lot of similarities with the viral mRNA. And they know of this. They know of this. The line 1 is old science, but they never investigated, or maybe they know something they're not telling us. And so I was just putting here that by a very simple PCR test, you could start screening for BioNTech, and if it were positive, you do sequencing. It doesn't, once again, it's just to show you the product, just to remind you what PCR is. So standard PCR test can do that. So if you're positive, it should indicate that you better start doing next generation sequencing to find out where on earth the copies have been inserted, because DNA was copied from RNA, and it's a unique proprietary sequence. So you can find out upstream, downstream locations. Wherever it was put in our genome can be found out today. It's just not being done. And so a lot of people, even if this particular PCR test was negative, it doesn't mean that there are other sequences from BioNTech or Moderna that were inserted. It's just that not enough research is being done. So there may be other sequences which are definitely only from the vaccine mRNAs that should be searched for, but nobody's searching for them because they don't want us to find out and answer these very, very important questions. So I'm just picking one target, but there are probably multiple targets for both vaccines. My wife's pointing at the clock, so I'm going to be done very, very shortly. So I'm basically saying that even if this PCR test were negative, it doesn't rule out other sequences having been inserted. And once again, this is a conceptual way of thinking about what sequencing can tell you. We have this about 1 to 2% of what we know here, the yellow box. And sequencing could tell you if it's inserted in where we know or if it's inserted in where we don't know. And it's important to know this. It's important to know this not only for that particular target, but for the whole rest of these mRNA vaccine sequences. And nobody is studying it. They don't want to. And this is just saying that our cells are very complicated with so many interacting components, enzymes, transcription factors, tumor growth factors, growth suppressors. Our cells we're very complicated organisms. Our cells are very complicated, and should only cancers be tested? By all means, no. I believe the miscarriage tissue from miscarriages should be tested. I believe our sperm from men should be tested if you want to have children, because those are rapidly dividing. And I was told by the research in Sweden, a lot of men came to her in her university saying, I want my semen tested, because semen, the dividing sperm, have high line 1 activity, and there's a tendency for this mRNA to find itself in ovaries, which they divide. The egg cells of women are born with, basically, but men's sperm cells divide a lot. And so there's a risk that because of the high line 1 activity in sperm, if someone's really concerned, why aren't they doing at least a simple start and try to see if the DNA is in their semen or not? Because that means it would be transmitted to the child, and the child will have it inserted who knows where, and you'll have children being born, with the DNA, because, you have the sperm and the egg come together. You only have one sperm and one egg. If that sperm has the DNA copies in it, it'll end up being the whole constitution of the child in every cell of a child. And none of this stuff is being explored. None of it. And it should be. And suppose experts, I understand, basic molecular science and all, and I've read refutations of the Lund study, and it's sort of like listening to the Inquisition discuss nuclear physics or something. These critics don't understand, they use sophistry, casuistry, and they obfuscate, but they never talk about actual laboratory testing that could rule this in or rule this out. They don't want to talk about actual testing that can be done in laboratories. We have the technology, we have the means, and we're going to have even better ways of doing things in the future. But right now, we already have what it takes to find out the likelihood of these messenger RNAs being copied into DNA and inserted into our genes. We have everything we need except the willingness to do it, and this would provide. You want to look at it materialistically, this would be a good way to make money for some people. And it would also employ a lot of geneticists and a lot of labs, because there's so much that has to be done. The safety of these things is not being tracked. Instead, everyone's just, when I went to medical school, I would not know that messenger RNA could be copied into DNA. We don't learn these things in medical school. It's because I learned that the PhDs and people probably smarter than doctors who have been studying this stuff when I was in medical school. I just never learned it. And so there's this wide open possibility that's pretty scary. And it's shameful that in our country we're not doing it. And that it took someone in Sweden to do it, and then she was shut down too. So, well, in any case, that's basically my message. And I apologize, I try to be as clear as I could, but it's kind of a vast subject, and the more I trip myself up, because the more I think about it, the more details there are that I want to address. So, but I'll stop here, and I apologize for going over time. Oh, one more thing. This is the fun part. So, this is just one thing. This kind of reminds me, here's the biblical message I have. Back around 1993, we were living in Maryland, and Rosita was doing her fellowship in genetics, and I was doing my fellowship in molecular pathology. And we'd often go, like almost every week, and we'd go to her parents' home. They live in McLean, Virginia. And, her mom would make a big meal and all that. And, her dad and I aren't Seventh-day Adventists. And one day we were having a toast. And it was shortly after Ceausescu fell from power, and he had power, and he bought this Romanian wine. He, like me, he'd buy stuff on sale, see if it's good or not. And so we were all, clinking our glasses, our wine glasses, over a bowl of salad, and my glass cracked. So a few little pieces of glass went into the salad. And I think my older daughter was already born. I don't remember if our younger daughter was born, but as a parent, I live in terror and fear of my children eating something sharp, broken glass, whatever, getting their guts ripped up inside their intestines. And so that glass of mine chipped over this bowl of salad. Her mom is bigger than that. Her mom made this beautiful bowl of salad, and I immediately said, we have to throw it out. And her mom and dad were like looking at me as if I were crazy. I said, no, we can't take a chance. We have to throw it out. I don't want anyone here swallowing glass, even though it had, olives and eggs and everything. It was beautiful, expensive bowl of salad. And so I went over to the garbage can, to everyone's dismay, but I think they all agreed with me when they thought about it later. I just dumped it in the trash. And this is what went through my mind back then, but I wasn't studying the Bible, but it applies. what was it, was it a scribe or a lawyer? I forgot. He said, Master, which is, in Matthew 22:3-6, he said, Master, which is the great commandment in the law? And Jesus said unto him, Thou shalt love Yahweh, your Almighty, with all thy heart, with all thy soul, and with all thy mind. This is the first great commandment. The second one. The second is like unto it, Thou shalt love thy neighbor as thyself. On these two commandments hang all the laws and the prophets. So, we've been living through something like a broken glass, a glass over a bowl of salad where some tiny little splinters somehow ended up in the salad. And we're being told to eat the salad. It's okay. It's worth the risk. It's a moral obligation to eat the salad. I wouldn't want anyone to eat a bowl of salad that might have even a tiny splinter of glass in it. I believe in throwing the whole thing away. I don't believe in gambling with people's lives. And yet that's what we've been told to do. At least we didn't do it consciously because we didn't know the full science. But the people we trusted know it. And they're telling us to eat the salad with the broken glass. And as far as I'm concerned, if you got a salad with even a splinter or two or wherever it might be, and there's a minuscule chance you'll eat the broken glass, no matter how expensive that salad is, I say throw it out. I say you throw it out and you do your homework first, and you hold people liable and you get rid of the indemnification, so that if anyone's hurt and you could prove it down the line, there's accountability in the Bible. There's no such thing as indemnification. You hurt someone, there has to be some kind of retribution or compensation, some kind of punishment, and I don't mean cruel and unusual punishment, but people have to be held accountable. And people have to pay out, not the taxpayer, the people who lobbied to make all this stuff mandatory. So that's what I wanted to say. And I'm open to any questions. And I'm happy to forward anyone who's interested in references, pretty good articles that are fun to read as far as I'm concerned. So thank you very much. Thank you.
Thank you, Dr. Paul.
Thank you.
Question? Any questions?
Oh, sure. Mm-hmm.
Basically, two questions.
Mm-hmm.
How do you think AI would maybe help in your pursuit of these
Well,
facts?
actually, I didn't.
Let me do
good
the
question.
second one.
Yeah.
And the CRISPR.
I'm
How
glad
do
you
you think of
mentioned
the
that.
CRISPR too?
I'm glad you mentioned both. And let me start with CRISPR first. You know how everyone's saying how you need the synthetic mRNAs and the nanoparticles containing them, and that's the future of medicine. And they're talking about cancer vaccines now. They're not vaccines. What they're doing is looking for what a given person's cancer might express that's abnormal, that their own immune system can attack. That's really immune therapy. It's not a vaccine, but they're trying to make us dumb us down so we use the same vocabulary in the wrong way. And so you asked about CRISPR. Well, I'll get to that. The problem is if you use these nanoparticle mRNAs, they're not necessarily targeting where they're going to be inserted. But CRISPR is a beautiful technology, not to make a vaccine, but to target where you want to insert a gene sequence in someone where it's worth the risk, someone with a terminal cancer. If you can break down a tumor activator, or you can activate a tumor suppressor by adding in sequences, or kill off some enzyme in the cell that's to slow down its metabolism. You could do that with CRISPR. You don't need nanoparticle mRNAs to do that. Like a skilled surgeon, CRISPR technology can insert exact sequences where you want them to go. And it's not perfect. That's why they shouldn't do it on fertile eggs before they turn into human beings. A lot of things could still go wrong. But in someone with cancer, yeah, someone with some kind of hereditary deficiency, which is normally deadly, and if you can insert a gene that'll cover for the deficient gene, of course. And CRISPR would do that so much better. You don't need nanoparticles with mRNA in them. We have a much better evolving tool that somehow they don't like to talk about because they invested so many billion in nanoparticle mRNAs. Oh, and the AI question, thank you. You know where the AI would be useful? I told you just how long our nucleotide sequences are, how vast they are. Like I told you, if you take each one, a nuclear base pair, that little ladder I showed you, and it was to a scale of 1 millimeter for every base pair, that would be 7,000 times the height of the Empire State Building. That's where AI comes in. AI could sift out looking for the sequences and where they're inserted. And this already is going on. You have programs where you can put in a given sequence and say, tell me wherever you find it and what's flanking it. That's AI, and it's already available, and it'll get better. It'll get maybe faster in terms of computer speed and all that, but we need AI for this kind of purpose, not for monitoring our social scores and our faces, but for life-saving medical information like this. Yes, AI is definitely tremendously valuable in this scenario and for the type of laboratory I'm talking about. And CRISPR technology is vastly superior to this nanoparticle stuff, which are just trying to make money on their investment. Very good question. And well, I hope some of what I said registered, because I know it's kind of vast, you know. And oh yeah, I can always give it again if you want to. I mean, yeah.
Okay.
Yeah, I'm sorry. I'm used to
Okay,
speaking to
Paul,
a
a number of us got the COVID-19 vaccine, Moderna or Pfizer. What are the risks that we incur?
I wish, and I'll take your question too. This is a good question. I'm going to take all of them. That's a very good question, David. And we don't know. That's the thing, because the research isn't being done. The laboratory testing is not being done. Instead, we're all being dismissed as I don't know what, conspiracy theorists, anti-Semites, whatever. This needs to be determined. Yeah.
Don't forget, he said that the most susceptible ones to get those cancers are those who have and the organs where you get these little cancers where your body is able to get rid of it on its own.
In theory, or people with pre-existing cancer, but it blows up after.
So there is a personal susceptibility.
Yeah, definitely, yeah.
So according to your lifestyle, your diet,
yeah,
and other things, maybe spirituality also, because it's God who heals us. So not everyone is susceptible, but it's true, there is the danger of that DNA going into our genes, but also don't forget that our body also has the ability to take out the wrong bases, nucleotides.
Yeah.
And so the healthier you are, the better your lifestyle, the higher you have a chance for your body to correct itself. So a lot of people, as he said, were those who had maybe small cancers or unknown cancers, so they get that, and boom, within a few weeks or months, suddenly they get this prominent cancer, young people sometimes. So our bodies, we are Adventists, we have a health message, our bodies does correct itself, okay? But of course it's better not to get the inoculation, but when we get it, of course, our body can fix it. So it depends on different people. Again, it's individual.
Thank you, that's good.
Two things. One is
I'll get to everyone.
Two things. One thing I saw yesterday, they had morticians getting together. They said they found a 30% uptake in white blood cell or blood clots in cadavers, and they've now upped their prices because it takes twice as long to embalm somebody.
Oh, I know what you're talking about. Yeah,
Okay.
It's a separate topic, but I'm happy to talk about it a little. Yeah.
Now, the other thing is NAC, they said NAC gets rid of, when you take NAC, it gets rid of about 12% of the spike protein out of your blood system. We'll get out that's in your organs, we'll get out 12%. Well, 5 weeks ago in Italy, they started selling augmented NAC. And it gets 99.8% of the spike protein out of your blood system. And I gave it to one of the members here, and his auction went from 85 to 100%, multiple times. So that one has been proven. It reverses in thousands of people. It's reversed long COVID. So it actually dissolves the spike protein and you eliminate it. So if people have symptoms, they should seriously looking to get augmented NAC. When I bought it, you can only get it from Italy. Now it's available in the United States from many sources, but it's like a brown glass bottle with a gold seal, but it's called augmented NAC.
Mm-hmm. No, I think that you're right to mention that. And I was, yeah, I'm not sure what it stands for, but I know there are a lot of treatments being started now. Mm-hmm. Oh, nicotide adenite, something maybe? I don't know. I don't know.
Some kind of vitamin B. It's possible, yeah. I'm not 100% sure. I have to Google it, but I don't know exactly what it stands for. Well, the more you eat your, remember, those vitamins are taken from what in laboratories? Of course, they can be synthesized, but also they can be taken from natural sources. So the more you have your raw fruits and vegetables, the more of all these vitamins you get, or your whole grains. I think if it's nicotinamide, I don't know what NAC stands for, but you can get it from your diet and from sunshine.
Oh, by the way, nucleotides and amino acids come from our diet. We can make nucleotides, and
Yes.
sometimes we can make some amino acids. I'm just saying that because nobody ever talks about where the nucleotides come from, so that cells get enough of them when they divide. But we get nucleotides in our food because plants even have nuclei with, genes in them, and so. And also, I just wanted to come back to a question that David was asking about risk. Keep in mind that I think probably for most people who get these injections, even if there is RNA being copied and inserted into our genes, it's a bit like a crapshoot or a roulette, because so much of our gene sequence is still not fully understood. It might be important or less important. It might end up in a place that isn't risky. And so someone might just be lucky, and whatever copy is made, if that's the case, will be put somewhere less important and won't have any effect on the patient. Oh, yeah, N-acetyl L-cysteine. Oh, I don't know much about that. Yeah.
It's the one
Yeah.
treatment during pandemic that
Mhm.
government made illegal to get, but you can get it now.
And there are other, like, what's it called again? I'm not a clinician and I don't prescribe drugs, but there was the one with the V that they were making fun of, and vitamin D and zinc and everything that we should do. They were saying. Oh, and you had a question too. Do you have the microphone? Oh, yeah. Oh, I mentioned that.
Can you elaborate on that, please?
Oh, yeah. And my numbers might not be accurate, but you know how we have 23 pairs of chromosomes? Well, I remember reading about this a while ago, and it fascinated me. But just like we have 23 pairs, the mustard seed could have something like 30-something, and not pairs, but copies in quadruple. And from species of mustard to other, some could have like 25. And sometimes, like, instead of 2 copies, 4 copies, and it's weird. And they have various numbers within this mustard. What's it called again? Species or something, or phylum. All these different mustard plants have wacky number of chromosomes, wacky number of line 1 activity. They have line 1, line 2, line 3 activity. And plants are pretty wild. You'd think that we, being the most advanced, mammalian creature, would have the most complicated and most copies of everything than anyone else, and we'd have more chromosomes than other species. But even the mouse, if I'm not mistaken, forgive me because my memory's a little rusty, I think they have more chromosomes than we do. But I know plants are wild, especially the mustard plant, having, some have 40 chromosomes, some have 50, some have 60, and wild stuff I can't even begin to understand, but I found very fascinating when I learned, when I was exposed to that. Mm-hmm.
You're concerned about the COVID-19, concern about the COVID-19 vaccination, was it as concerning as the previous vaccines, the annual vaccines?
Well, no, and I
the-
used to not care about any of these
of
things.
the flu virus, of the
Yeah,
flu vaccine.
yeah, well, I never got them, I never
Right.
got any of
But
them
is
because I
there a
didn't believe.
parallel between this
Well, the
traditional
difference
annual
now
flu viruses
they were different mechanisms. The so-called old-fashioned vaccines were proteins mostly.
Right.
Proteins, maybe a range of proteins to cover the immune. You get a few antibodies against a targeted virus, and they were proteins that didn't stay in you forever. They were injected into you, and your immune cells, coordinated with each other to start making antibodies against the proteins. This is a completely different technology, and they always say it'll teach your cell to make the antigen to make the antibody against. I mean, given what the Nobel Prize was given for, I would say it would fool your cells into taking in foreign, alien, long-life messenger RNA to hijack your cell to get them to make a protein that it shouldn't be making. Yeah, very different.
Okay.
Yeah, I mean, I had no problem with
Thank
the
you.
old-fashioned
Right.
vaccines, none
I didn't know if
whatsoever.
there's any parallel.
No. I have no problem with the old-fashioned vaccines personally. I mean, there are other problems which are usually more like autoimmune responses and stuff like that, problems with the excipients, but they weren't teaching your cells to make anything. They were injected in you to create an immune reaction, and that comes with its own fair share of problems, but not to the extent that these messenger RNA injections created. I mean, there are problems with some of them. I'm not saying those are perfect, but I used to not even think about it because I think you inject someone with proteins eventually, gotten rid of. Here, they're teaching your cells by injecting them with sequences, gene sequences, and an mRNA is a gene sequence. And it's not a human gene sequence, it's a multi-purpose sequence that, like I said, has these open reading frames like a viral mRNA that's not our usual mRNA. A human mRNA, usually what they call an open reading frame, is making just one protein, not multiple proteins on the same mRNA. With the BioNTech, with the virus, and with the Moderna, they have all these open reading frames which are possible that can be, under certain circumstances, create other proteins other than the intended protein. And why? I don't know. And I can't find any answers in the literature. And given that they understand ORF1 and ORF2 for so long, why can't they at minimum give an explanation, and why can't they test for insertion of copies of their patented genes? Do you know how the gene sequences were discovered, or at least for nanotech? Someone on some research team had to go through medical waste to find a vial, and they used the leftover in the vial to run the sequencing on, take the steps necessary to run the sequence. I had to go into the trash to find it. And I don't know if Moderna gave it voluntarily, but for a while neither of them wanted to give their sequences, and they said it was proprietary. Oh, yeah, proprietary, well, who's paying for the property?
Yeah.
$16 trillion wasted.
Okay, real quick, you mentioned DNA was copied from RNA. Can you explain that just briefly?
Yeah. Line 1 has two components. The ORF1 protein binds the messenger RNA and chaperones it into the nucleus. And it's thought that it's probably when cells are dividing, when they're most penetrable by the proteins, that's when the membranes have more spaces. So it's believed that the open reading frame 1 protein, they both bind to it, but the open reading frame 1 sometimes binds in more copies, and it chaperones, it transports the messenger RNA into the nucleus, and then the ORF2 component copies it into DNA and inserts it in a way right into the DNA, so it splices it, and you have symmetrical copies. You just have a new gene, basically, or copy of a gene or part of a gene.
So what he's trying to say
Yeah.
is that they told you that the RNA does not turn into DNA because normally a DNA is copied into an RNA. The DNA is the master gene, all right? But the problem is when the mRNA goes into the nucleus, the nucleus itself has this ORF1 and
Well,
all these
ORF1
things.
brings it into the
Yeah.
nucleus.
So
And-
your own cell is able to take that mRNA, turn it into DNA, and then insert it into your genes.
Yes.
So that's in very, very simple language.
And the guy who won the Nobel Prize stated there's no risk for mRNA. He didn't prove it. They didn't test, they didn't do anything to prove that it isn't possible. They did nothing, zero, or they didn't publish it. Because I looked all over. It's all shut down. Thank you. Oh, I'm sorry, I'm done if you want me. Yeah.
Thank you so much for attending this presentation, and
Thank you for having me.
thank you, Dr. Paul, for
Oh,
coming
thank you,
and
thank you.
sharing your experience with
I
us.
appreciate it. I appreciate the opportunity very much.
Let's have a prayer. Our most gracious, loving Heavenly Father, we thank you so much for your Sabbath day. Thank you for your love. Thank you for your health message that just given to us by Dr. Paul. Help us to learn what we hear and apply into our daily life, and prepare ourselves ready for your soon return. In Jesus' name, amen.
Amen. Thank you, David. Thank you.